be on redefining drugs to combat gastric cancer pathogens, prospects and purpose by our renowned speaker, Dr. Nagendran Dharmalingam. He is a good friend of mine and my missus, and he is right now doing his postdoctoral research fellowship at the Department of Medicine under the Division of Infectious Disease and in the Alpert Medical School of Brown University, Rhode Island. And to give an information about his education, he completed his BSc Microbiology at Bharatidasan University and went on doing his MSc Microbiology at Bharatidasan University by the year 2010. And he got his doctoral degree awarded by the year 2015 at Yonsei University, South Korea, in biomedical science. And his research interest is focused on antibiogram of microorganisms, different microorganisms, and different antimicrobial resistance. And he is trying to address the facts that are related to novel antimicrobial molecules. And he is also doing research in infection biology. And his current research focus is on gastric cancers and finding an alternative medicine, redefining the alternative medicine and different types of drugs. And he has procured about 7 awards. Notably, he has secured the BK12 Korean Government Scholarship Award and Outstanding International Grad School Scholarship Award in South Korea. And to his record, he has around 23 research articles in both national and international research journals, and all are highly renowned and applauded journals. And he has also filed a patent, US patent, which is an international patent pertaining to the cancer biology. And his other activities are like he is involved in, he's a live member of American Society of Microbiology, which is an esteemed group, a scientific forum, which discusses on different aspects of microbes every year and provides an important solution to the world. And he also served as a president and secretary of Rotaract Club and Students Club during his time in India. And he is also an editor in various journals, and notably he is an editor in Omics group of journals, and also he is also doing a reviewing job, a research journal reviewing job, a journal of medical, like many journals, notably Journal of Medical Microbiology, so on and so forth. So with these few informations, I would like to invite Dr. Nagendran Dharmalingam to deliver his speech on gastric cancers. And I would say this is a topic relevant to current scenario because the current lifestyle has increased the percentage of this gastric cancers. Almost all different types of cancers getting increased the percentage, but notably gastric cancer is one of the phenomenon which is highly getting increased due to change in the human lifestyle. And there was a report which I went through this morning by WHO, which states that it was a report published by the year 2003 or 2004, but then it has signified, by that year it has signified that by the year 2020 around 50%, that is 500,000 people around the globe would be suffering from cancer, that is gastric cancer. And so finding a drug that could minimize this effect is really an important thing in this current scenario. So I hope we would get a briefing or an insight into what is going to happen in the cancer biology, especially in the gastric cancer biology. So let's hear from what Dr. Nagendran has to say. Please welcome, sir. Thank you.
Thank you, Dr. Srinivasan. This was wonderful. An introduction. I'm really happy about the introduction, and thank you for the opportunity that you are providing, and I'm really honored to present the details, the data, what I have, and which will be helping for the students getting a short intro about the novel antimicrobials and then repurposing. So here I would like to, let's start that, and I'm going to present window. Can you see my screen?
Please, let me check. Yeah, it's visible.
It's visible? Okay. Once again, thank you for this opportunity. Let me give you, thank you, Dr. Srinivasan. He gave me a very fantastic introduction about me. I'm really happy about the introduction. And basically, I address myself as a medical lab technician. And then I'm honored to be a medical, I'm honored that I'm saying that I'm a medical lab technician. So basically, I started my career as a medical lab technician. And then I did my bachelor's and master's and my PhD, and I did my PhD in medical lab technician or medical laboratory science. So I mean, basically I'm a paramedical staff. So then turned into a scientist, and now today we are going to discuss about the drug repurposing. So we have various terminologies about how to reuse the drugs, which is currently using in the clinical in the clinical side. Um, we have lots and lots of definitions like drug repositioning, repurposing. So most of, as you can see through my screen, to find a different or to find a challenging other target of a drug which is reported and using for the different purpose. Uh, right now we are just tuning the purpose. That's what we are looking for. And my focus here, drug repurposing, how a drug can be used in a different manner. Like it is starting from identifying and developing and to commercialize, which is existing or abandoned drugs. So by using a high throughput screen, like we use different robotic arms to screen the drugs, which is in a high throughput manner. So we ourselves in our lab we screen like 85,000 compounds. From that screen we found like 200 165 hits, as we call hits. Based on the hits, we scrutinized and then we shortlist some compounds which can be used. That's what we call developing. So after identifying by using a high throughput screen, we are developing assays to getting into and then to addressing and then to know about the properties of the drug, and then in order to commercialize the drug which is in the existing or abandoned drugs. Abandoned drugs, just we can say like a drug that can be forgotten or that can be not prescribing due to the low penetration effect or tissue penetration effect or some other side effects kind of stuff. That we are giving a rebirth, real life to the other drugs by using drug repurposing strategy. Let's getting into the drug developing. As you all hear of university, Tough University, which is one of the very famous and then a very high reputed university in the Boston. There is a center called Drug Development Center for Study of the Drug Development. They found a single drug which is cost about 2.1 billion. If you convert into Indian currency, it's thousands and thousands of crores rupees to develop a single drug. Can you believe? So, and then the cost of a drug which is increased over the years, over the decades, as you can compare, the graph is a very inclining phase that you during the 2000 and 2010, and now this is really doubled. It should be doubled. That's why the FDA, what we call like that is what we call Food and Drug Administration, one of the drug policing and a drug regulating agency here in this country, USA, they prefer or they just highly recommended to repurpose the drug. And the reason why the success rate is really high success rate compared to the novel drug. As you can compare a novel drug, we don't know anything. I mean, a researcher or investigator who dealing with a new drug, he didn't know anything about the efficacy and efficiency, the pharmacodynamics pattern, everything. We didn't know anything. However, the clinical already approved clinical drug, we know all about the details. All we need to just tune a little bit of based on the target. Based on that, the success rate will be really high, 1/10 of success rate. And then the safety, obviously, there is lots and lots of factors that that influencing the toxicity of the drug that can induce that can induce various other side effects in the host. Hence, the known safety, which is the highly recommended and positive factor to develop a drug which is in a clinical use. And then the other important factor, so a single drug which is at least 12 to 20 years is needed to develop a drug, what we call a bedside to bedside, simply a simple term which is called bedside to bedside. A bedside, the term what we use to develop, we start identify and develop a drug in the lab, and then to bedside, the drug which is useful and useful to prescribe to the patients, that we call a bedside. So the time span that what we call like 12 to 20 years. However, when we go for the repurposing stuff, it's like 1 to 3 years we can finish everything. And I can give you a best example of the drug repurposing here right now. Yes, the hot topics, and the hot topic is SARS-CoV-2. The organism, the virus which is causing COVID-19 disease. And as you all hear of these kind of chloroquine and hydroxychloroquine and chloropromazone, the drugs, doxycycline, all these drugs, you kind of hearing this kind of stuff in recent days. That is what we call a repurposing. And when you go to the website clinicaltrials.gov, that will show you what are all the current ongoing research on repurposing the drugs that will be using for the new virus or new disease, new infectious disease, or new other disorders, something like that. And when we let's take an example. Let's discuss about the SARS-CoV right now. There is a many number of drugs that is ongoing research on. It is initiated. The chloroquine drug, what we know, it is a simple medication that is a cheap medication using for the malarial diseases, and also the other drug called montelukast, that drug which is using for the treating the asthma patients, and also the chloropromazine, that drug which is used for treating the psychotic disorders, and then another doxycycline, just a simple antibiotic. So that antibiotic which is known for to inhibit the bacterial RNA synthesis. However, here this drug which is they're proposing to stop the cytokine storm, that is the main culprit of the fatality causing by the viral infection. So this is the best example right now, and why we need this repurposing. And as you can refer from the previous slide, the time duration which is very less, and as you can see this, the remdesivir, one of the drug which is initially they invented for the Ebola disease, and now they are prescribing for the SARS-CoV. So that's why we need these kind of fast approvals, that we need drug repurposing. The strategy is really useful, and then time saving and cost saving, money saving, and patient saving. That's the most important thing. These are the manuscripts that we published in our by our research group in our lab, anti-helminthic drugs, niclosamide, oxyclonoside, and ornofin, one of the anti-inflammatory drug, and then diflumin is one of the anti-inflammatory drug, and also niclosamide. So these and then I contributed 4 papers in our lab. So that's why I'm interested totally in repurposing. Today we're going to talk about a drug repurposing strategy to combat the bacterial infection, and then we will shortlist. We'll discuss more detail into what is the exact pathogen what we're going to see about. So my topic here, my research, major research we are discussing, we called escape pathogen. So as we can see, the first letters of all the pathogens is called ESKAPE, escape, C. That is class in BPC, one of the addition after it entered the list. So these are a hospital-acquired pathogen. When a person who admitted in the hospital and undergone any surgeries or procedures, a catheter insertion or kind of other procedures, they're having a high chance of getting infected from this pathogen, what we call hospital-acquired pathogens, what we call escape pathogens. the most of my research, they are focusing on to combat these pathogens, and also the other pathogen called gastric pathogen known as Helicobacter pylori. I'll tell you the interesting story later. And then some intracellular pathogens, Francisella tularensis, Staphylococcus aureus. Believe it or not, Staphylococcus aureus is also one of the intracellular pathogen. So this Francisella tularensis is called biobar weapon, that you may have heard of. Anthrax bacillus anthracis bacteria, which is the causative agent of anthrax, one of the very famous disease where the people are using during the Cold War season kind of stuff. So let's focus a little more about H. pylori, and then why we need to focus, why we are against H. pylori. According to the World Health Organization, according to the World Health Organization, the priority pathogen list was disclosed. I mean, they published in terms of to find a new antibiotics, which is a critical need. And as you can see here in the priority 2, Helicobacter pylori clarithromycin resistance. So that antibiotic resistance H. pylori, clarithromycin resistant H. pylori, which is emerging pathogen that will be highly dangerous to the human population. As you can see here from the left side of the right side of the screen, the global population which gonna see about 10 millions of death because of due to antibiotic resistance, and then it will cost a very big money, that is called 100 trillion because of this antibiotic resistance. And about in 2050, the people will die. People will infect, or people will face the antibiotic resistance, which is higher than the cancer. So right now, the cancer death mortality is the higher number in this right now, but it will. I mean, the antibiotic resistance will overcome the numbers. So that's why we all need a new antibiotic. And then I'm focusing on Helicobacter pylori, which is my cup of tea. And yeah, what is Helicobacter pylori? Simply, we can say ulcer bacteria. That's what I can say. I mean, I believe that most of you students, you're all from bachelors and masters. A simple terminology, I can say that ulcer bacteria. A person who got an ulcer, probably at most he got a H. pylori positive. The Helicobacter pylori is a gram-negative pathogen, and then which is the helical shaped spiral shaped bacteria, and also it need it is a microaerophilic bacteria that doesn't need too much of oxygen like other bacteria requires. um, it can survive in your stomach, almost like 50% of human population harboring this pathogen. You want to believe this, but that's true. That's what I mean. The bacteria that is known as type 1, class 1 carcinogen, that is classified by the World Health Organization around 1994. This bacteria, oh, see how beautiful the bacteria is. So the bacteria having the bacteria was discovered by two scientists, doctors, physicians called Barry Marshall and Rabin Warren. So these fellows, they're doctors in I think the name of the hospital is Victoria Hospital in Australia. Let me tell you a very interesting story about this finding the bacteria. Actually, Barry Marshall is a physician. I mean, a junior physician joining the hospital. He was working under Robin Warren, and then he was seeing gastritis patients. I mean, too many numbers of gastric patients. The patients, and then he believed that why the gastritis can be caused by, or can be a reason, a bacteria can be the reason for the disease. So he started isolating the gastric samples and then start to culture on the plates. However, he didn't succeed. Mm, everyone was laugh at him. Everyone was laugh at him. So that's why most of the scientists, I mean, the scientists are inspiration to most of the people. Uh, Everyone's starting initially like they all are laughing at you when you are doing, or when you're trying something. However, they start believe you when you achieve That's what happened here in this case with the Barry Marshall and Rabin Warren. Everyone was laughing at, oh my god, how this gas it is, how this ulcer that can caused by a bacteria that can serve, how it can survive in the gastric pH. As you know, as you can imagine, the gastric pH is 1 to 2. I mean, the pH is 1 to 2 in the in the stomach region. However, they believed. I mean, Barry Marshall, he believed himself. I mean, he believed his stuff. He believed his knowledge. He started culturing his bacteria. It's not growing, but he's not succeed. However, he's keep trying. One fine Good Friday. Actually, we all know that we are having holiday on Good Friday, and then it's all on the Easter Sunday. So he having 3 days of holiday when he, when he cultured the bacteria on Thursday. So he cultured the bacteria on Thursday and put the plates on the CO2 incubator, and then he forgot. I mean, he left home like after 3 days or 4 days. On Monday, when he come to the lab and he observed the plate, yes, there is a bacteria in the plate. I mean, the bacterial colony in the plate. That means this bacteria, which is the slow growing bacteria, which need at least 3 days to grow. That's how he found the bacteria. That's why we say that most of the scientific inventions are accidents. Like we found antibiotic, like as accidental finding. That's how the Helicobacter is also accidental finding up to me, because it is intended to see after 1 or 2 days. However, he forgot the plate in the incubator, and then he found the bacteria in the plate that after 4 days. However, that's not the end of the story. He want to prove that bacteria, which is the causative agent of ulcer. You know what he did? He just drunk the bacterial culture. He drunk the bacterial culture, and then after a few days, Barry Marshall's mom, she was Barry Marshall's mom, she smells very bad breath from Barry Marshall. So he asked, "Hey Barry, I'm sensing very bad breath from your body. So just go and check what has happened." Then he was imagined that it should be because of the H. pylori, and then he went to endoscopy. And he confirmed he developed ulcers. And then he got after he confirmed the ulcers, he took antibiotics. He recovered from the infection, and believe it or not, he got a Nobel Prize in 1990. I mean, this is one of the very fantastic story. How it is impressed me. He tested himself. So scientists, they believe. I mean, who believe themselves, they will become a good scientist. So Barry Marshall, he believed himself, and then he became a scientist, and he became a Nobel laureate. That's how he got the bacteria. I mean, that's how he got the Nobel Prize. The bacteria which is present in the stomach, I mean, as I said, is 50% of the human population. They have the bacteria, and then when you have kind of gastritis infection, I mean gastritis kind of symptoms, please go to the doctor, and then cure immediately. That's the most important. I mean, that's the most important advice as I can say, as a H. pylori expertise I'm developing myself here. The amoxicillin, clarithromycin, and any one of protein pump inhibitor is the basic regimen of this bacteria. However, nowadays the bacteria is so brilliant than us, so it is developing resistance. That's how we are doing our job here. We found the interesting report that is called repurposing niclosamide to combat H. pylori. So niclosamide is one of the drug which is an FDA approved drug that used to treat tapeworm infections. Like நம்ம வந்து நாடா புழுன்னு சொல்லுவோம், like நாடா புழு. Like decades before, 20 years before, it was a very big infection. I mean, very big problem in our country, like all the tapeworm, pinworm, roundworm infections. So like that, the tapeworm will grow like it usually grows like 1 to 2 meters in length. This drug is a miracle drug for this tapeworm infections. That's how initially they found this niclosamide is anti-helminthic drug. However, we got a good information that repurposing niclosamide that can inhibit the Staphylococcus aureus growth, and my finding is it can inhibit the H. pylori growth. Before going to repurpose, so what is the main strategies, or what is the main the bullet points that we need to remember to to go before, before we pursue efficiency of the drug, which is so important, though. How it is efficiency, how efficiently it inhibits the bacteria. That is the first important thing that we call. In addition, we can say that in other words, minimum inhibitory concentration in terms of bacterial inhibition. And then it should have a potential mechanism of action. It should kill the bacteria. It should kill the bacteria. That's all the next thing we have to focus. And then it will be added advantage if it is synergized with the other clinical antibiotics. So synergize means that it could be it could be useful, or it could be when it combined with other antibiotics, it could act more efficiently than it alone treatment than it treated alone. So this will be the other character of, if it is synergist, it will be really a good advantage of the drug we are developing. And then it should resist, and also it should resist to the antibiotic resistance. As you know that bacteria developing resistance, which is one of the brilliant mechanism. So a drug, it should resist to developing the antibiotic resistance. So that's what I can say, that resist to antibiotic resistance, that should be the other important category. And then it should pass the toxicity screen. That most of the, the FDA approved drugs, they all having the informations of toxicity, and then we can tune a little so to become a good antibiotic candidate. And then it should be efficacy in vivo, that it should rescue a host, like a mice or rabbit or rat. It would rescue the infection. So these the three, six important factors we should consider before we can pursue the antibiotic. So when it comes to the efficiency of how it inhibit the bacteria, so the MIC should MIC is like, the niclosamide MIC was 0.25, which is less than the other metronidazole and other antibiotics. However, which is little higher, which is higher than the common generally used antibiotics used for the H. pylori infection. However, why we pursued this antibiotic, we have the justification, good reasons. I'm going to discuss later in the slides. So when it comes to the minimum concentration, you guys all know that how this assay going to do. So we simply dilute the molecule in a different concentrations, and then we add the bacteria in it. Simply, at what concentration the compound should inhibit the bacteria, which is known as minimum inhibitory concentration. As you can see here, the green color indicates the bacterial growth, and then the orange color indicates no growth. So we initially dilute the compound, gradiently, twofold diluted the compounds in the test tube, and then add the bacteria in it. So after 24 hours or 48 hours or 72 hours, after the prescribed incubation time, we took the bacteria and then see visibly what is the maximum, what is the minimum concentration inhibit the bacteria. So that's how we report the MIC. And as I said before, how a good molecule should synergize with the other antibiotics. So likewise, our niclosamide candidate, which is really a good synergized molecule against omeprazole, metronidazole, pantoprazole. Those are the proton pump inhibitors. And then which is pretty good with clarithromycin amoxicillin. Fortunately, we not develop any antagonistic effect. That is, when it combined, the activity which is not decreased. That's what we say that antagonism. So that's how we choose this drug, which is one of the can be a potential candidate. And the killing mechanism, what we can say that the bacteria at high concentration, as you can see here, at the 10 to the power of 8 number of cells per ml concentration, the drug which decrease at least 1 CFU. However, we can say that bacteriostatic molecule in high concentration. When how we found this assay, that when there is an increase in concentration of antibiotic and increase in time, so the percentage of survival of the bacterial cells decreased. At one point, the concentration of a molecule will completely kill the bacteria that we usually sees that 99.9% of killing effect, as we can all see in the advertisement. So that's why we call the bacterial killing mechanism that bactericidal effect. However, our bacteria, which is a bacteriostatic at high concentration of bacteria, but why we focusing this molecule can be a good potential molecule. See the acidic stability. As you can see here, the clarithromycin which increase, I mean the MIC is increased. In other words, the efficiency is decreased when there is an acidic pH. However, our drug which pertain and the one, there is no fold change when there is a low pH or acidic pH. That means when a drug which consumed through our stomach, it should survive, and then the mechanism of action should sustain at that constant, I mean at that pH. So that's why we choose that. Okay, this may be a good candidate because it survives stable in the low pH acidic pH. And also, after 30 days of continuous supply of this antibiotic, it didn't develop any resistance up to 30 days of passaging. However, as you can see here, a fourfold increase when there is a clarithromycin. That's why the World Health Organization, they found that the clarithromycin resistant antibiotic, clarithromycin resistant H. pylori, which is the one of the emerging pathogen. As you can see here, there is a emergence of and resistance against clarithromycin, which is crystal clear against this bacteria. So there is no resistance. However, you can see twofold resistance, twofold increase in MIC against amoxicillin. It also known as I mean also emerging thing. So that's how niclosamide, our drug repurposing stuff, I mean repurposing drug, that can be a good potential candidate. And also it decreased the bacterial motility. So how the bacteria, it penetrates the gastric mucin gel above the gastric layer, the gastric gastric epithelial cell layer. And as you can see, the mechanism how it's spirally shaped, and then it goes inside the finding the epithelial cell and attaching on the cells. As you can see here, this is one of the H. pylori motility, that how it spiral shaped, it going through towards the cell. That's how I mean our drug at low MIC, that is sub MIC level, it can inhibit the motility of the bacteria. That's what we can see. That's what we saw. And then next thing, uh, after penetration of gastric mucin layer, it can target or it can go towards the gastric epithelial cell, and then it adheres. Attachment is the preliminary step of colonization. When there is a colonization, the adhesion is the primary critical step. And then, as you can see here, this is a model of gastric epithelial cell. When after penetration of gastric mucin layer, I'm sorry, the bacteria goes toward the epithelial cell, and it adheres on the surface of the epithelial cell and goes into the gastric epithelial cell, and it induces the signal transduction pathway of the signal transduction pathway that will lead to the disease, either gastritis or it is ulcer, either intestinal metaplasia that leads to a gastric cancer. So our drug, it potentially decreased the bacterial, how we did this assay, that like once we infect the gastric epithelial cells with H. pylori, we leave it for some time, and after that we washed off the unattached bacteria, and then we treated the compound. So these are everything happening in the cell culture dish, as we can see here. So it's kind of a model epithelial cell, and we can add the bacteria, and then it attach on the surface of the layer, and then we treat the compounds after treatment. So we see the CFU in other words colony forming units, that how many of colonies, how many colonies, bacteria present in the infection. So as you can see here, when there is a comparison with the amoxicillin, so niclosamide potentially decreased 1, 2, 3 CFUs. So which is really a good number, which is really exciting about our results. And also it clears the intracellular, that is invaded bacteria. So that drug can goes inside the epithelial cells and kill the bacteria present inside epithelial cells. That's what we can see. That's what we call it invaded bacteria invasion. So this bacteria, I mean this drug, which is what we can see here in the gastric region, can be acting as a good candidate on also it decrease the inflammatory cytokine that is called IL-8 secretion. That is called inflammatory response. It decreases the inflammatory response. So, like we are seeing, we got a good results about the in vitro mammalian cell assays. Um, these assays, as you can see here on panel A and B, it's a normal bacteria, untreated bacteria. However, as you can see, the C, D, E, F, G, H. So the CD panel represents a bacteria which is treated 8x concentration of its MIC, and as you can see here, the total morphology has changed uh, after the bacterial treatment that indicates. So when there is a morphological change, that also results in a decreased host pathogen interaction that will lead to rescuing or recovering from the disease condition. And IJ panel, which is the control antibiotic amoxicillin, as you can see here, the difference between the panel AB and then IJ and KL, those are the control antibiotics. So when comparing to the standard antibiotic amoxicillin, our drug also potential in clearing the infection in the means of decreasing its, changing its morphology, surface morphology of the bacteria. And then the infection as I referenced, the bacteria which is having a two important two important toxins, which is known as cytotoxin associated antigen CAG-A and VAC-A. When the gastric epithelial cells, as you can see here, the panel A, which represent a normal epithelial cells, like as you can the enlarged version. This is not the exact infection as we did, but I'm just referring the other manuscript to making it clear about the picture of mammalian cells. This is a normal mammalian cell. If you have a look at the panel C, that shows a small round vacuoles present inside the mammalian cells. That represents infection results. After the infection, and then the vacuolation results in after H. pylori infection. However, in the panel D, it doesn't have any single vacuole. As you can see here, there is no other vacuole. But however, this epithelial cells infected with H. pylori and treated with niclosamide. So the in vitro data also showing that a potential candidate of niclosamide is a potential candidate, not only inhibiting the bacterial growth and also inhibiting the signal transduction mechanism, which is caused by the H. pylori bacteria. And here you can see here the VAC-A. This is a bacterial cell decreased bacterial cell membrane. Bacterial cell that was treated with H. pylori. Bacterial cell treated with the niclosamide that decrease toxin transcription. So in other words, the bacteria the infected bacteria, after it treated with niclosamide, it decreases vaccine transcription and translation thing that leads to the decreased toxin entry into this cell. So what we're seeing in this, obviously, good results that it inhibit the bacterial vacuolation. So the mechanism of action is also one of the very important thing that we need to know. Every antibiotic should know about. We need to know about the mechanism of action. So when it comes to a clinical antibiotics, as of now, what we have practicing here, practicing a different kind of antibiotics that focusing on 4 or 5 main targets, that it inhibit the bacterial DNA replication, or it inhibit the bacterial mRNA transcription, or inhibit the protein translation, or other, in other words, the antibiotic that can inhibit the bacterial enzymatic activity. That is one of the critical to make it bacteria to survive. However, our drug that play with the bacterial plasma membrane proton motive force, that is decreased. I mean, that is the proton motive force is decreased by the niclosamide, as you can see here. Panel B, I mean, the panel C, it is having the, I mean, there is no difference in the treating kind of thing after it. 100% potential of the bacterial cell, bacterial membrane. However, when we treat with the bacteria, the bacteria with the niclosamide, as you can see here, there is a decrease in the bacterial membrane potential that can be compared with the panel A, which is one of the known known antibiotic called valinomycin that results after treatment, that results in bacterial membrane increase in bacterial membrane potential. So as compared to the positive thing, we can see that there is a decrease in membrane potential results in bacterial membrane damage. So that's all about the mechanism of action and cytotoxicity. I mean, basically the cytotoxicity is a little higher. I mean, the toxicity is higher in the gastric cells and epithelial cells. But when it redefined with the other study by the chemistry, I mean, with the association with the medicinal chemistry, it should be decreased. That's what I'm seeing here. The other model, what we are calling like wax moth model, infection model. So we took this larva of this wax moth that is called like Galleria melanola. We took that worm, and then we infect the worm with the bacteria. That results in if it is a pathogenic bacteria, it will die. So after infection with bacteria, we infect with the drug, which is mimicking a mice study. So mice is different; it's a vertebrate model, but this is the not invertebrate model that we took the Galleria melanola wax moth larva, and then we inject the bacteria and also with the niclosamide. As you can hear, a very statistically significant rescuing of this. From the bacterial infection that we can see here. So based on this all assays, we are seeing this. The niclosamide is a low toxicity. I mean, the whole toxicity is just less than the other thing, because the niclosamide is the drug which is low absorbing capacity of this drug results in very less absorption throughout the system. However, what are targeting the human gastric region, that is only covered by the drug that can be utilized on the surface clearing of the bacteria. And then we filed a patent on this niclosamide. We have fingers crossed. We are assuming that it will be happen soon. And then we are in stage of initiating the clinical trials that will be dealing with the direct patients. We need to recruit the patients to see the efficacy, and then finally, as I told you before, the repurposing strategy, which is a time, cost, and effort saving. Still, there is a big room is waiting to you guys to open. So after finishing your course, getting into the research and explore the unexplored drugs that can be using for the new purpose. So I sincerely thank my late Appa and then Amma and sister wife. So because of them, I'm here, and I would like to thank my postdoc mentor Lafir Mailnakis and other colleagues, and another my guru Marudamuthu Sundaram sir. So he was a principal scientist where I did my masters. He taught me a research, how to research and what to do in the research, So he is my guru. I really thank him, and then obviously my PhD guru, Professor Kim Jong Bae. He is a person, only because of him I successfully finished my PhD. So, and I thank all the department members, faculty members in where I did my masters and the PhD. Obviously, and the family and friends. Because of them, I'm here, and the colleagues and collaborators. And thank you very much for your kind attention. And if there is any open for discussion, if you have any questions, feel free to ask me. Thank you very much. Thank you.
Yeah, thank you, sir, for that nice presentation. Gratitude
Yeah,
is an important quality of human being, and the way you acknowledged each and every person in your life shows how far you have transcended your path, and you are right now at the current state of position. And we wish, on behalf of our institution, we wish you all the very best in the future endeavors. Participants, if you have any questions, you can unmute your mic and you can discuss with the guest. They have also typed few questions.
yeah, Saiha, I saw some questions.
Yeah.
So, can you please elaborate on the combination therapy of H. pylori in terms of mechanism of action on the PPI? So, PPI, like proton pump inhibitor, as you know that our gastric region, I mean our stomach, which is able to produce the hydrochloric acid that is resulting into digest our food what we are taking. However, a few drugs that cannot be stable or that can be stable in the gastric pH. That's why the drug clarithromycin is designed. So the clarithromycin is a drug, one of the macrolide antibiotic that is discovered. I mean, they developed the drug in order to stable in gastric pH. However, still it need to be. I mean, the pH should be. The acidic thing should be decreased. So that's how we take proton pump inhibitor. That will decrease your pH, and also the combination of clarithromycin and amoxicillin that will result in potential inhibition of this bacteria. That's what I'm seeing here. That's the main mechanism behind the synergistic pathway.
Thank you, sir.
Welcome. When small dose of same type of cancer-causing virus or bacteria injected into cancerous patients, is the antibody are produced, or how the antibody react to the cancer antigen. So the question by G. Sarita, she was talking. I mean, her question is a really interesting question. Like a small dose of cancer-causing virus or bacteria can be injected. So most of the cancers, I mean, all the cancers are different in the manner how it is initiated, how it progressed, how it's metamorphosed, and then metastasis happen, and how it is circulated to a human. If you talk about prostate cancer, the gastric cancer pathogen that cannot be helpful in prostate cancer, and the hepatic cancer that cannot be helpful in this kind of stuff. So here, Helicobacter pylori, let's say gastric cancer initiating either infectious pathogen, either a bacteria or virus. The toxins produced by the bacteria that play with the signal transduction mechanism in the cell where it adhere to, and then it tweak a little. I mean, it switch a little. That will result in a single cell change. That will result in a cancer cell on progression. Let's talk about the H. pylori KAGA protein. Now, the KAGA protein, when it get, I mean, when you talk about a bacterial protein injection system, there is a lot. There is a various secretion system presenting outside of the body, presenting outside of the bacteria, that is called type 1, type 2, type 3, type 4 to type 8 or type 9 secretion system. Through that secretion system, and the bacterial protein injected into the human gastric epithelial cell, that protein, after getting entry into the human epithelial cell, it will play with the beta catenin, one of the transcription factor that induce the translocation of the beta catenin pathway that goes into the nucleus, binding on the oncogenic genes that initiate the oncogenic mechanism. So here, the gastric cancer is totally important. The antibodies producing the H. pylori against the H. pylori or virus, that is totally different. So long story short, the cancer-causing pathogen, injecting the pathogens, antigens that will develop. Obviously, they are developing antibodies that will give you a resistance against the disease. I mean, the infectious condition that will not ever. I mean, it will not like potentially inhibit the cancer initiating mechanism. Obviously, you can defend your infection, not with the cancer thing. So that's all I can see here. Yeah, obviously. I mean, the next question, irritable bowel syndrome. I mean, there is a bacteria called Bacillus. Oh my God, I'm forget that. It's not coming. Oh God, I'm Bacillus fragilis. So that bacteria, one of the anaerobic bacteria present surviving in your stomach, in your stomach region, that will result in induce the irritable bowel syndrome and Crohn's disease and etc. So nowadays, people, they are relating everything with the bacteria. Still there is a page to explore. Everything they are relating, you know. And nowadays, the people, they are relating your gut bacteria to your thinking, your stress thing, what we call like gut microbiome axis. How your gut bacteria induce or induce with the will be induce your thinking or your thoughts of mind. So it can be it is happening. Um, does the aminomycin does the immune system resist the bacteria, at least in some of the people? Yeah, you're right. Some geographic region people, they are resist to some kind of bacteria. Say for an example, let's talk about the H. pylori. So when the Helicobacter pylori, which is characterized at East Asian type and Western type, based on the phosphorylation pattern of the GAG protein. So most of the people in most of the region in East Asia, they are having the East Asian type, and people in the Western countries, they are having the Western type. So based on the geographic region and how the bacteria which is present from the birth, it can be like uh, uh, proposing thing that people in different region, they have the immune, like they can resist certain type of bacteria. I'm sure about that, sir. In spite of many herbal medicine, high level research in cancer nowadays. Okay, yeah. Sumaya Rahman, Sumaya Rahman talk about the herbal medicine cancer treatment. Let me tell you very interesting thing. நம்ம தமிழ்ல வந்து ஒரு விஷயம் சொல்லுவாங்க, 10 மிளகு இருந்தால் பகைவன் வீட்டிலும் சாப்பிடலாம் அப்படின்ட்டு. Which means I did my PhD in black pepper. If you go type my name, you can find the inhibitory effect of piperine on Helicobacter pylori infection. So that is the first report ever recorded against H. pylori in the worldwide. No one ever published a report on piperine, inhibition of piperine against H. pylori. So when you take a certain amount of piperine, black pepper every day, it can the ability to inhibit your gastric H. pylori bacteria growth inside your stomach. That was proved by my reports that. அதனால வீட்டுல எல்லாரும் சொல்லுவாங்க, வீட்டுல எப்பவுமே டெய்லி ரசம் சாப்பிடணும்னு சொல்லுவாங்க. So our rasam it's a herbal juice. It contains most of the active ingredients that can inhibit the gastric cancer thing. So I'm doing my research on medicine, on the chemistry medicine side, I support our herbal medicines. So the most important thing is, please don't do any crude extract research. Don't do any crude extract. Just isolate a single compound and see the effect of single compound. That is most important. People, most of the people here, at my era, they are doing this blunder, like doing a crude extract and tested and then reporting. Don't do this, please. So if you're getting into research, taking a single medicine, I mean single compound, that will be results in fantastic results. So I reported like inhibitory effect of I mean in antibacterial and anti-cancer, anti-inflammatory effect of piperine against H. pylori. So who knows? The doors are opening for you. So just go and find the right place and do your good work. And Divya Meenakshi, she did PhD in piperine. Okay. If you have any question, I mean, if you have any interesting thing to discuss, I'm happy to just welcome and discuss with you all. And thank you very much for your information. It's 19 money I was very tired.
And thank you, sir, for such a wonderful clarification. If you have participants, if you have any more doubts, you can contact him through mail. We will share his email ID. And once again, thank you all for participating in this lecture. And special thanks to sir for being here with us today. And
thank you.
Naga Chalti to you. Naliki da ungalukku. Okay. Anyway, advance.
And அனைவருக்கும் இனிய விநாயகர் தின நல்வாழ்த்துக்கள். நன்றி.
Sure. Let me share the feedback of today's session. Let me share the link.
Yep.
So participants, please find the link attached. You can click on it and you can answer your feedback. And you can also find the email ID of Dr. Nagendran. You can contact him for any sort of queries. Feedback, once you complete it, you will get your certificate through your email. Well, thank you, sir.
Oh, thank you very much.
Thank you.
Take care all. Good night.
Good night. Participant, the platform will be open for another 10 minutes. You can complete your feedback and submit it, after which the platform will close.
Hi, I'm Brad Cowan, and in this video, I'm going to show you how anyone can quickly and easily